Photochemistry would be messy if light were not quantized.
Science Makes Me Smile, presented by young Dr. JGC, a creation of old Dr. JGC.
Light quantization is essential for vision. I am young Dr. JGC. Let's begin. Before we begin, let us define light quantization in very simple terms, for those of us not trained in physics. Light quantization means that light does not deliver energy in a smooth, continuous flow, like water from a faucet. Instead, it comes in tiny, discrete packets called photons. Each photon carries a fixed amount of energy determined by its color or frequency, and this energy cannot be divided into smaller pieces. In other words, light behaves less like a steady stream and more like a series of tiny identical energy packets, each capable of triggering a specific change in a molecule if it has exactly the right amount of energy. Light quantization is essential for how molecules like retinol interact with light in biological systems. Retinol is a small light-sensitive molecule, yellow molecule in this picture, located in the photoreceptor cells of the retina in the eye, where it is tightly bound inside a protein called opsin. Together, they form rhodopsin, the molecular system responsible for detecting light. Opsin is a transmembrane protein, purple in this illustration. In this partnership, retinol acts as the light sensor, while the protein converts that event into a biological signal. Retinol possesses discrete electronic energy levels, for example, a ground state E0 and excited states E1 and E2, and a transition can occur only when the energy difference between two levels is exactly matched. When retinol absorbs a photon with the correct energy, one of its electrons is promoted from E0 to E1. In this picture, blue electron, ground state, red electron, excited state. This electronic excitation triggers an ultra-fast structural change. Retinol flips from a bent 11-cis shape to a straightened all-trans form. This transformation acts like a molecular switch. A helpful analogy is a spring-loaded latch. Retinol sits in the protein like a bent key under tension. When the right photon arrives, it releases the latch, snapping retin into a new shape. That tiny mechanical change pushes on the surrounding protein, causing opsin to shift its structure. This shift initiates a cascade of biochemical signals that ultimately become a nerve impulse, what we perceive as vision. Because light is quantized, this process is remarkably precise. One photon activates one retinol molecule, producing a single, well-defined switching event. This one photon, one match transition, one mechanical response relationship makes vision extremely sensitive and reliable, even in very dim light. Quantization also prevents what could be thought of as an energy overdose. Retinol cannot gradually absorb arbitrary amounts of energy. It either absorbs a photon with the correct energy, or it does not absorb at all. If light were not quantized, energy could in principle be transferred continuously, allowing the molecule to accumulate excess energy in an uncontrolled way, potentially leading to erratic or non-specific changes. Without quantization, energy transfer would not align with these discrete electronic levels, and the precise switch-like behavior of retinol would be lost. In such a world, the controlled, reliable reactions that underpin vision and photochemistry more broadly would give way to unpredictability. In other words, without quantized light, photochemistry would be chaotic.
Now you can see how the quantization of light allows vision to be both precise and highly sensitive. Fascinating, isn't it? Let me know your thoughts in the comments. And as always, thanks for watching Science Makes Me Smile.